AITA. Am I the apoptotic for being the most important enzyme in photosynthesis, but also kind of bad at my job? All right, listen carefully. I am Rubisco. Yes, that Rubisco, the enzyme you all talk about but never seem to appreciate. I feel the need to set the record straight because, frankly, I'm getting an undeserved reputation. I am the most abundant enzyme on the planet. Without me, photosynthesis wouldn't exist. No photosynthesis means no sugars, no plants growing, and frankly, no life as you know it. I am essential to the carbon cycle that sustains every living thing, from the smallest microbe to the largest mammal. Let me be clear about where I fit in all of this. I am the key player in the Calvin cycle, the process within photosynthesis that actually turns carbon dioxide into sugars, the building blocks of life. My job is to capture carbon dioxide from the air and attach it to a 5-carbon sugar molecule called ribulose 1,5-bisphosphate, RuBP for short. This step is called carbon fixation, and it's the very first and arguably most important step of the Calvin cycle. Once I do that, the cycle continues, eventually producing glucose and other sugars that feed plants and, by extension, all of life on Earth. Now, I won't deny that I have my flaws. I'm not the fastest, and by some measures, I'm considered inefficient. Occasionally, I make the unfortunate mistake of binding oxygen instead of carbon dioxide. This process is known as photorespiration, and yes, it is often blamed on me. When I bind oxygen, the plant produces a molecule called 2-phosphoglycolate, which can't be used in the Calvin cycle to make sugars. The plant then has to expend additional energy to process this molecule, transporting it to other parts of the cell, such as the peroxisome and mitochondria. This cleanup requires ATP and NADPH, precious energy the plant could otherwise use to grow or produce more sugars. In short, I cause the plant to waste energy correcting my mistake. Moreover, every time this happens, valuable carbon and oxygen molecules are wasted, forcing the plant to backtrack and expend even more resources to compensate. It's a frustrating inefficiency, I admit. Now, I'm not without competition. Over time, some plants have developed clever strategies to work around my inefficiencies. C4 plants, like corn and sugarcane, have evolved a way to capture carbon dioxide more efficiently by physically separating the initial carbon fixation from the Calvin cycle. They use an enzyme called PEP carboxylase, which doesn't make the same oxygen binding mistake I do. This allows them to concentrate CO2 around me, dramatically reducing photorespiration and saving energy. Then there are CAM plants, cacti and succulents, for example, that take a different approach. They fix carbon dioxide at night when it's cooler and store it until daylight, when I'm ready to use it in the Calvin cycle. This adaptation helps them conserve water and reduces the chance that I'll bind oxygen instead of carbon dioxide during hot, dry days. These adaptations highlight just how challenging my job is. The fact that plants have developed such elaborate workarounds shows that I'm far from perfect. Yet, despite my shortcomings, I remain the cornerstone of photosynthesis for the vast majority of plants on Earth. I understand that some refer to me as the bottleneck of photosynthesis and argue that if I were more efficient, crop yields could improve significantly, especially under stressful environmental conditions. While that may be true, I am doing the best I can given the evolutionary constraints and the chemical challenges I face. So, before you dismiss me as inefficient or a waste of resources, remember this. Without me, the entire foundation of life on Earth would collapse. I may not be perfect, but I am indispensable. And if that makes me the flawed workhorse of photosynthesis, so be it. I am Rubisco, and I get the job done, even if it's not always pretty.